Good morning, good afternoon, and good evening, everyone, and welcome to today's webinar titled Neurovascular Coupling, Novel Insights from Studies in Awake Head-Fixed and Anesthetized Mice. We are joined by Dr. Adrián Rodríguez-Contreras, Associate Professor in the Roxlin and Richard Pepper Department of Communication Sciences and Disorders at the Northwestern University School of Communication, and Dr. Barbara Lind, Assistant Professor in the Department of Neuroscience at the University of Copenhagen. They'll be discussing their research on neurovascular coupling, including two-photon imaging and optogenetics, and the effects of anesthesia. I'd also like to extend a big thank you to our sponsor, Neurotar, for making today's event possible. Neurotar develops research instruments that facilitate imaging and electrophysiological recordings in awake head-fixed mice. They use their own equipment, the mobile home cage, for contract research and supply it to academic labs like Adrián's and Barbara's. Neurotar also share their protocols and help labs transition to awake preparations. If you'd like to meet the Neurotar team in person, you will find them at this year's upcoming SFN meeting in San Diego at booth 2036. So please drop by. I'm sure they'd love to speak with you. I'm Andrew Davies from the events team here at scientist.com, and I'm happy to be your host today. Before I welcome our first speaker to the floor, I wanted to cover a few brief housekeeping notes to help you get the most out of your experience. First, this webinar is being recorded and will be available to view on demand at some point in the future, so stay tuned for an email notification. For this reason, screenshots and recording during today's presentation are not permitted. Next, if you take a look at the interactivity panel at the right of your screen, there are 4 tabs corresponding to chat, Q&A, polls, and handouts. This is where you'll participate in the Q&A and polls during the webinar today and access all webinar resources. If you experience any technical issues, please first try refreshing your browser, as this fixes most problems, but if you're still having trouble, please reach out using the private chat function and send a message to me at Inside Scientific Events Team. We'd also love to hear from you, so please send any questions, thoughts, and comments using the Q&A tab, and we'll try to address as many of your questions in a Q&A session following the presentations. If we don't have time to address your question today, we'll forward it on to Adrián and Barbara afterwards. And finally, you can also explore relevant resources in the Handouts tab to learn more about some of the research presented today. Just before our first presentation, we have a couple of quick polls for you. The first question is, which of the following research methods do you currently use? Please select all that apply, and the options are two-photon imaging, electrophysiological recordings, behavioral assays, optogenetics, functional ultrasound imaging, and wide field imaging. We'll just give you a moment to submit your responses, and again, please select all that apply. Okay, thanks very much. Next, if you currently work with awake rodents, which of the following do you use? And again, please select all that apply, and the options are full body restraint, head-mounted electrophysiological probes, other head-mounted imaging devices, commercially available airlifted ball and or treadmill, custom-made airlifted ball and or treadmill, or not applicable for those of you who do not work with awake rodents. And again, we'll just give you all a moment to submit your responses, and please select all that apply. Okay, thank you very much to everyone in the audience for your participation. It is now my pleasure to introduce our first speaker, Adrián Rodríguez-Contreras. Adrián, take it away whenever you're ready.
Okay, so hello everyone. My name is Adrián Rodríguez-Contreras, and today I will tell you a series of recent studies that got started with a collaboration with my dear colleague, Dr. Lingyang Shi, who is at the bioengineering department at UCSD. And so what I want to do is I want to first acknowledge my colleague, and also indicate that what I'll be doing today is just give you an overview of these studies. But if you are interested in more details, you can look at the paper that has been, the preprint that has been uploaded in bioarchives. So the first thing to point out is that Our work is focused on the cerebral vasculature. And it is, as many of you know, the cerebral vasculature is essential for brain function because it provides oxygenation, nutrition, and protection to brain tissue. It has also been evident that dysfunction of the cerebral vasculature is involved in a variety of conditions that range from inflammation and stroke to neurodegenerative diseases such as Alzheimer's. And finally, the fact that the cerebral blood flow and cerebral metabolic rates are coupled provides the basis for several non-invasive technologies used to image the brain in humans. In more recent years, there has been tremendous progress in the characterization of the cerebral vasculature from a microscopic viewpoint, highlighting the cellular constituents of what is known as the neurovascular unit. So this slide shows a little bit more detail. So imagine that you slice the human brain and then basically begin to look at the distribution of different vessel types in the tissue. And a key component, of course, of the vasculature is the endothelial cells, which form the actual blood vessels. But there are a number of other cellular components that are very important. For example, in this diagram or in this cartoon, the smooth muscle cells shown in green and the pericytes shown in cyan are key cell types that associate with arteries and penetrating arterioles and with capillaries. And they are important for the control of blood flow because they are contractile cells. Okay, so constricting the blood vessels will reduce blood flow, and relaxing these cells will enhance blood flow. And shown in pink and black are the parenchymal cells, astrocytes, and neurons, which of course have interesting functional interactions with blood vessels. And the mechanisms of intercellular communication between the cell types are under intense study by several groups. Now, of interest to us is the fact that general anesthetics, which are used for surgical procedures in clinical settings and in preclinical studies, have very broad effects on blood flow and metabolic rate. For example, isoflurane is known to increase blood flow and to decrease metabolic rate, literally uncoupling this important relationship between these variables. And so this volatile anesthetic is known to cause vessel dilation, but how it affects a decrease in metabolic rate is less understood. So this slide also shows that there are different mechanisms or different processes that regulate blood flow in the brain. There is the process of cerebral autoregulation, vasomotor reactivity, and the process of neurovascular coupling, which are acting at different levels of the process in different vessel types, and but all of them involve an interaction with these mural cells that are contractile. So how we want to address this is basically we know that isoflurane induces a dilation of vessels, and our research question is, what is the effect of perturbing vascular tone on calcium activity of cerebrovascular endothelial cells? So to approach this question, we have taken an in vivo approach. We take advantage of mouse that is available through the Chromus, the Cornell Heart Lung Blood Resource for Optogenetic Mouse Signaling. And these animals have a genetically encoded calcium indicator that is under the control of the CDH5 promoter, which is very active in endothelial cells of all the body, but also in the brain. And this allows us to measure calcium activity with this optical sensor. And then the approach that we use is we use we implant a head plate on these animals, and we perform a thin skull preparation, which consists in simply thinning the skull. In this particular case, what we did is we used stereotaxic coordinates to locate the location to find the somatosensory cortex. And then we don't remove the skull, but we thin the skull, and we use this preparation then to image the activity of the calcium indicator in the endothelial cells with a 2-photon microscopy. Here's the setup in the slide, indicating that we are using femtosecond laser to excite the calcium indicator and then image its activity while animals are awake. And then we have in the setup a nose cone that can be retracted and positioned close to the snout of the animal for gas delivery. So this is the gas delivery protocol that we are using. We have a baseline where the animal is breathing air, the animal is awake, moving, and then we introduce the vehicle, which is oxygen. And then what we do is we apply isoflurane in a condition that resembles what goes on in many laboratories around the world. There is a stage of induction of the anesthesia with 5% isoflurane, followed by a period of about 10 minutes of anesthetic maintenance. And then at the end of this period of maintenance, we let the animal recover by breathing regular air. So here is a series of videos that I want to show in three different time points of this protocol. The first one is the baseline in air. The second is during the induction of with 5% isoflurane, and the third one will be the recovery stage. So if you bear with me, I will play the first video, which shows the calcium activity that we observe in these animals under baseline conditions. So here you can see that there seems to be some vasomotion in this particular field of view, and there are several events. And now what I'm going to show is the next video, which is now when we apply isoflurane. And so what you see is that very quickly the vessel at the center of the field of view has dilated, and the calcium activity has decreased over time. And then lastly, I'm going to show a video that shows the situation when we let the animal recover. So here you can see that there's a long time going by, and then the vascular tone returns to what looks like a constricted state, and very slowly activity comes back. So what we've done is first look at the vasodilating effects of isoflurane. And what we've done is we classified the image vessels into three groups. We have the small diameter vessels, the medium diameter vessels, and the large diameter vessels. And we didn't observe any evidence of vasodilation in the small diameter vessels, which have vessel diameters of around 10 microns. Here is the average and standard deviation of the diameter of these small vessels on baseline, induction, and recovery. But we saw evidence of vasodilating effects in medium vessels and in large vessels. And interestingly, we didn't seem to see a return to pre-induction diameters in these two vessel types. let's have a look a little bit more carefully at the different vessels. And what I've shown here is how is, let's say, the time course of change in diameter. For small vessels, we don't see a very consistent effect of isoflurane. Now note that the diameter scale is different for each vessel type, but it's simply to highlight the changes in diameter that take place after we apply isoflurane. And so you can see that, for example, most of medium vessels very rapidly begin to increase in diameter, right? Within a few seconds of isoflurane exposure, and within 50 seconds, they are fully dilated. And the same situation is for large vessels. So basically what I've shown so far is that isoflurane dilates medium and large diameter vessels, but it doesn't seem to have a clear effect on the small diameter vessels. What about the calcium activity in these different vessel types? Well, what I've done here is show a summary, picking up some exemplary traces under baseline induction and recovery condition for small vessels, medium vessels, and a large vessel. And you can see, for example, that under baseline, there are different levels of activity in these different vessel types. And during the induction, there's a very interesting time course, which begins with an increase in activity followed by a slow decrease in activity for all of the different types. And somehow the kinetics of this rise and decrease in activity seem to be different, slightly different for the different vessels. And there's a much slower time course of recovery of the activity in all vessel types. What we've done is we normalize the change in fluorescence for the three different vessel types. In cyan is shown the small vessel types, in green are the medium vessels, and in red is the large vessels. And you can see that here is the time course in bins of 50 seconds. You can see that the time of half maximal inhibition is different for the different types, but it's around 100 seconds for medium and large vessels, and it's much longer, around 200 seconds for small vessels. So what we are interested is in beginning to address whether there is a correlation between changes in vessel diameter and endothelial cell changes in fluorescence with this optical reporter. And we try to address this in two steps. The first step, what we did is we used multiple regions of interest along vessels of interest. In this case, for example, there are the green regions of interest are shown for a medium-sized vessel, which is in very close proximity to a large vessel. And we are comparing the same number of regions of interest, and we are going to compare what's going on in the change in fluorescence in the entire frame. And then what we did is, in a second step, is begin to look in more carefully at these regions, these whole tracts of vessels, and look at changes, correlated changes in area and fluorescence. And so what we find is, I think is very interesting. Here shown in pink is the trace of the whole frame, and in red and green are the individual regions of interest of these two different vessels. And now I'm showing you all the different steps in this gas delivery protocol. So in baseline, what you can see is there seems to be synchrony in these events that we are recording in individual vessels. And this correlates very well with peaks in the frame change in fluorescence under baseline and vehicle conditions. And interestingly, during the induction condition, there seems to be a synchronous rise in activity in these multiple regions, followed by a decrease in activity. Now, here is an observation that it becomes a little bit of an anecdotal situation, that there seems to be an increase in activity very closely associated when we start imaging in these different conditions. And what we have done in the maintenance situation is show that the activity, the decrease in activity is sustained, but there is no rise that is correlated with the start of the imaging. And the reason I'm saying this is because there is, for those of you who are familiar with two-photon imaging, there is a shutter that comes on and makes a click noise during the start of image acquisition. And we were worried that what we were triggering is this synchrony, right, through sensory stimulation. But the maintenance condition shows that this doesn't happen. So what is interesting is that there are some small events happening during the maintenance condition that seem to be uncorrelated with each other. And then when we have the recovery condition, very slowly the calcium activity returns, and the synchrony of the events seems to get back. So what we found in the next step is that we found a correlation between changes in the area of the vessel and the fluorescence. And so on the left side, there are the correlation coefficients for small diameter vessels in the center, for medium diameter vessels, and for large diameter vessels on the right side. And if you focus your attention in medium and large diameter vessels, what we found is significant positive correlations between these two variables under baseline conditions, but that during the induction conditions, they drop to non-significant values, and then very slowly sort of begin to return to high correlation values, but then remain in the non-significant correlations even after recovery, suggesting that these vessels where we see strong dilating effects of isoflurane also have disrupted synchrony and correlations between changes in vessel size and fluorescence. And now, in terms of what's going on with the small diameter vessels, we found some evidence that the correlation between these variables actually begins with non-significant values and then increases, right, to significant values during the maintenance of a condition, and then return to non-significant values during the recovery condition. So what I've shown so far is that isoflurane dilates medium and large vessels, and we don't see a dilating effect on small diameter vessels. And we also don't really know if these are direct or indirect effects on the neural cells, but certainly it could be something that could be addressed in future studies. We have shown that during the induction of anesthesia, there's a rise in calcium activity in endothelial cells. And here, of course, we want to speculate that this fast rise has to do with the effect of isoflurane. Probably could be a direct effect on endothelial cells, but it could also be an indirect effect on other cell types. We have also shown that there is a longer-term decrease in calcium activity during the induction, and that during this time, there is a disruption of calcium activity synchrony that is sustained during the maintenance of the anesthesia. And I want to point out that last year, Longden and colleagues published a really interesting article where they begin to look at the mechanisms that generate calcium events in endothelial cells. This is a study that used the same reagents that were used in our study, but in their case, they were doing very detailed manipulations in anesthetized animals. So I highly recommend that you look for this article, and this could be a further follow-up for people who want to initiate a conversation about these results. So our current hypothesis is that there is a feedback mechanism between intracellular calcium fluctuations in endothelial cells and the maintenance of vascular tone. And we are proposing that we can use anesthetics as reagents to perturb this relationship. Lastly, I would like to acknowledge our funding seed support, which is essential and is allowing us to take on this new line of research. And thank you for your attention. I'm happy to answer any questions that you may have.
Thanks very much for that great presentation, Adrián. And just before we move on to our second presentation, we have our last two polls. And our first question is, which statement best describes your current situation? The options are, I currently work with awake rodents and am interested in head fixation techniques. I will work in awake mice in the future with optical or functional ultrasound imaging. I will work in awake mice in the future in electrophysiological studies, or I just have a general interest in the subject and technique. And I'll just give everyone a moment to submit their responses. Okay, great. Thank you very much, everyone. And our last poll for today is, which of the following behavioral readouts would you like to combine with imaging or electrophysiology? And please select all that apply. And the options are visual, auditory, tactile stimulations, locomotion tracking, spatial learning, reward-driven learning, maze navigation, open field exploration, novel object recognition, social interaction and memory, passive and active avoidance, or other. And again, please select all that apply. Okay, thank you everyone for your responses. They're very much appreciated. And it is now my pleasure to welcome Barbara Lind. Barbara, the floor is yours whenever you're ready.
Hello everyone, my name is Barbara Lind, and today I would like to present a project that I've been doing studying PV interneurons in awake mice and how they evoke astrocytic calcium responses that may contribute to the neurovascular coupling that we see. Yes. As Adrián presented so nicely, there is blood flow delivery in the brain, and in the barrel cortex where I work, the blood is delivered through the penetrating arterioles that branch down from the surface of the brain, from the arteries on the surface of the brain, and spread into the capillary bed, which then joins the penetrating venules that brings the blood back up to the surface. So there is a constant delivery of blood, but in neurovascular coupling, this blood is locally increased dependent on increased neuronal activity. And this is a signal which is used to study human brain activity in the fMRI. And typically, we consider this blood flow response to be dependent on increased neuronal excitation. But besides the excitatory neurons, there is of course also a bunch of different inhibitory neurons in the tissue. And several of these inhibitory interneurons have been shown to induce a blood flow response in what you might call inhibitory neurovascular coupling. So many inhibitory neurons will release vasodilatory compounds that can act directly on the contractile cells on the blood vessels. But some, like PV interneurons, mostly or only release GABA. And GABA, currently we have no explanation for an effect directly from GABA on the blood vessels. So a suggestion could be that this GABA works via the astrocytes in a way similar to what have been described with glutamate-based neurovascular coupling, because astrocytes will release vasodilators calcium dependently. When it comes to astrocyte contribution to neurovascular coupling, it's of course markedly obvious that the astrocytes cover the blood vessels via their end feet, both on the penetrating arterioles and on the capillaries. But it has for many years been debated whether the astrocytes really contribute in vivo to neurovascular coupling, because many studies have seen that astrocyte calcium activities will occur later than the dilation of the blood vessels. But this is, of course, also dependent on which astrocytic calcium activity one is studying. With regards to inhibitory interneurons, astrocyte calcium responses have been found in both slices and in anesthetized mice, but they are typically described to be slow or fluctuating over a long period after the activation of the interneurons. And such activity is, of course, also hard to reconcile with a contribution to neurovascular coupling following increased activity of these inhibitory interneurons. Finally, with regards to these controversies, there might be some explanation in the findings that astrocyte calcium will contribute to dilation of capillary, but not arterioles. So it's very important that you are aware which part of the vasculature you are doing your investigation. And in our experiments, we studied the neurovascular coupling in awake mice through a chronically implanted cranial window. And the first thing we would do was to follow the flow in the superficial blood vessels to identify where the penetrating arterioles were, and then following them into the tissue. The setup we had was allowing the mouse to move in this neurotar floating pad while we were doing the imaging, and its head was fixed on the objective of the 2-photon microscope. And we visualized astrocytic calcium activity using virally introduced GCAM6F in 2 different mice strains, one which would express channel rhodopsin under the VGAT promoter, and one where the channel rhodopsin were expressed under the PV promoter. And then finally, we visualized the lumen of the blood vessels using Texas red dextran, which was given IV just before every imaging session. And you see the Texas red here in the lumen of the blood vessels, and the GCAM6 in the astrocytes. That is the green signal. So this is what it looked like when the mouse is moving around in the neurotar stage. And we have the 2-photon imaging of this preparation, and we are in layer 2 of the cortex, around 150 micrometers below the surface. And you can see that astrocyte calcium activity really rise dramatically when the mouse is running. which has been shown many times before. But we also see that there is a lot of activity going on in this awake mouse independently of the running activity. So in this context, we did our experiment stimulating either just the PV interneurons, which would release just GABA onto the astrocytes or the tissue in general, and then compared it to optogenetic stimulation where all interneurons are activated using the VGAT promoter. And that will, of course, influence directly the PV interneurons activity, because some inhibitory neurons inhibit the PV interneurons. It will also give rise to a release of numerous other compounds, such as a big arsenal of neuropeptides, and also, for example, the vasodilator NO. And in this way, we could compare this more complex signaling to a simple GABA release from the PV interneurons. And we did that both in anesthetized and awake mice. So initially, we confirmed that our optogenetic stimulation will induce an inhibition of the tissue by placing an extracellular local field potential recording electrode into the craniotomy. And what we saw was that during our optogenetic stimulation, there was a significant decrease in the negative amplitude in our local field potentials, reflecting that, yes, we had indeed inhibition during our optogenetic stimulation. When we considered the astrocytic calcium response in the anesthetized mouse, we didn't see much. But then when we did the same experiment in an awake animal, we saw a huge calcium response, both after PV stimulation and VGAT stimulation. And this was the case both with the shorter and a longer stimulation of 15 seconds. And here we have the calcium response in astrocytes to a VGAT stimulation. And what you can see is that, first of all, we cannot image during our optogenetic stimulation in our setup, but as soon as the optogenetic stimulation is done, the calcium would build up in the astrocytes. And when you look at where this calcium response was taking place, it's occurring in many small regions all over the field of view. In comparison, here we have the optogenetic stimulation to a PV stimulation. And what you can see is that once the optogenetic stimulation is done, the calcium is already up in the astrocytes. And you can also see when you look at the structures as to where the calcium increase occurs. First, we have calcium increase in some elongated regions of the astrocytes, and then later there will be activity which is more central to what appears to be somal structures. And this later fluctuating calcium response appears to be very comparable to what other people have reported from slice works in astrocyte calcium studies. So these elongated calcium increases in astrocyte to the PV stimulation, when you look up in under G here, you see that we could see them very close to this the position of the interneuronal structures, possibly reflecting direct effect from a GABA release from these structures. Another point with regards to timing is that the PV-induced calcium activity seemed to be in the astrocytes seemed to be two-phased. So first, there will be an increase which would begin already during stimulation, and then there will be a second peak. And whilst the stimulation with the VGAT based optogenetic stimulation, that will give rise to just one calcium peak in the astrocyte and a little later, which is reflected, of course, in a significant difference in the levels just after stimulation between the two different stimulation paradigms. and also in the time to peak. So somehow this mixed stimulation of the VGAT-based of all the inhibitory interneurons actually delays the calcium responses, or only recruit part of the astrocytic calcium response that we see when we stimulate the PV interneurons alone. So astrocytic calcium activity has been shown to depend on noradrenaline levels. So we tried to modify the noradrenaline levels in the mice using a neurotoxin called DSP4. We took out the locus ceruleus based noradrenaline release, and what we saw was that this reduced reduction of noradrenaline would reduce primarily the first part of the astrocytic calcium response to the PV stimulation. So in summary, we have a clear difference between how astrocytes will respond to either of the two optogenetic stimulations, depending on whether the mouse was anesthetized or awake. And this may, with regards to the PV stimulation, be actually dependent on sufficient noradrenaline levels. But of course, this may reflect that the awake state of the mouse and the sufficient noradrenaline levels makes the astrocytes more susceptible to GABA. But since we are working in awake mice, it's also possible that our optogenetic stimulation is inducing a behavior of the mouse, which is the basis of this increased response in the astrocytes. So first of all, we did not include any recordings in this study where the mouse was running either before the stimulation or during the stimulation, or for 20 seconds after the optogenetic stimulation. And we have cameras recording the activity of these mice that had been trained repeatedly to be included in the experiment. And as you just saw in this video that I started, this mouse is sitting very still during our first optogenetic stimulation and then the 2-photon imaging. And that is, of course, not always the case. This mouse, a different mouse, a different day, it's more awake, I would say. The other mouse was not anesthetized in any way. It's maybe sleeping, I don't know. But this mouse is definitely moving its whiskers and running a little bit. But as you will see soon, it's not very well correlated to the onset here of the optogenetic stimulation, and neither with the onset of the 2-photon imaging when that started. So, but to be sure that we were not eliciting a behavioral response with our optogenetic stimulation, we took the data that we had from the Neurotar pad that allowed us to track how much the mouse moved. And of course, we used that to exclude the recordings where the mouse was moving a lot. And then we also used a camera to focus on the whisking of the mouse, because it's possible that the optogenetic inhibition of the cortex could trigger the mouse to want to move its whiskers once the stimulation was over. So we focused on the whiskers, and as you can see, when the mouse is resting its whiskers, we see those whiskers very clearly. But when the mouse is moving its whiskers, on the picture to the right, there is a bigger diffraction of the images, and in this way we could determine when the whisker was moving. So either it's we classified a time period as either with moving whiskers, that would be 1, or without moving whiskers, that would be 0. And the red trace we have here is from when the mouse is moving a little bit. But based on this data, we could then find out if the whisking had increased after the optogenetic stimulation. And you see the black trace here is the average whisking in relation to a 2-second long PV interneuron stimulation. And we saw no significant change or increase in the whisking after the oxygenetic stimulation compared to the spontaneous activity at an unrelated time point. And below that whisking activity, you see the movement activity traced from the pad below the mouse, but obviously that would be 0 or close to 0 because we excluded the acquisitions where the mouse was moving. And this lack of whisking movement was also evident in longer PV stimulation of 15 seconds and in the VGAT-based interneural stimulation, both the short and the long ones. So with that, we could exclude that our stimulation was somehow changing the behavior of the mouse, and trust to a large degree that our activation of the PV interneurons was the basis of this increase in astrocytic calcium that we saw. So we went on to study the neurovascular coupling response, and in the anesthetized mice, where we did not have any increase in astrocytic calcium, we did not see a dilation of the penetrating arteriole to the PV stimulation, but we had a dilation when we stimulated based on the VGAT promoter. So we stimulated all the inhibitory interneurons. And that is probably due to the release of vasodilators directly from the inhibitory interneurons. When we then looked in the anesthetized, in awake mice, we saw that the blood flow response was much enlarged, both to PV stimulation and VGAT stimulation. And interestingly, the timing of these two blood flow responses does not seem to be identical, so that with regards to dilation, it's slightly slower following a PV stimulation than following the VGAT stimulation. So when we then compared these observations to the astrocytic calcium activity, we compared the timing of the peak of the astrocytic calcium activity and the peak dilation of the penetrating arteriole and the first order capillary, which is the first branch point from this penetrating arteriole. And we saw that specifically with regards to the VGAT-based interneuronal activation, the astrocytic calcium activity would peak later than the dilation of the penetrating arteriole and the first order capillary, which underline that in the context of a general activation of inhibitory interneurons, astrocytes cannot be an important contributor to the neurovascular coupling. But with regards to the PV interneuron, the picture was a bit less clear. But then we should remember that the astrocytic calcium signal to the PV stimulation had two phases. And if we did not look at just the largest peak, but considered the first of the peaks, regardless of its size, we would see that the astrocytic calcium would always increase before dilation in the PV interneuron stimulation, both of penetrating arterioles, but significantly with regards to the first order capillaries. And we then did a small correlation study in the way that first here D is with regards to dilation of the penetrating arterioles. We looked at the dilations where there was an astrocytic calcium response before and compared it to the dilations that did not have a clear astrocytic calcium response before the dilation. And those with the astrocyte response before, they are the green, that is the line with the green standard error of mean around. And the ones without the astrocyte activity is the one with the red SEM, and we don't see any effect on the first initial dilation, but it seemed like the dilations of the penetrating arteriole with astrocyte activity has prolongation in the dilation. But when it comes to that effect is not so strong, but when it comes to the first order capillaries, we did the same sort of separation, and we saw that the dilations preceded by an associated calcium response were much larger than those that did not have an astrocyte response before. And when we took the data from the mice, when the mice have been treated with DSP-4, so taking away the noradrenaline, we had seen that would take away the first peak of the astrocyte calcium. And here we saw that the dilation of especially the capillaries, but also of the penetrating arterioles, was reduced and was of size comparable to those dilations that did not have an associated calcium rise before the dilation. So with this, we could summarize that yes, that the PV inhibitory interneurons would induce an astrocytic calcium activity in the awake mice, and that this occur on a time scale that allows for astrocytic contribution to inhibitory neurovascular coupling. And our results also support that astrocytes are involved in GABA-dependent neurovascular coupling, particularly at capillaries. So with this, I would like to thank my small group, Jakob, Gabriella, and Aske, and the funding that had supported me so far, and you for your attention, and I'm happy to take any questions that you may have.
Thank you, Barbara, for that fantastic presentation, and thank you, Adrián, as well. We have a bit of time left, so let's move on to our Q&A. And I'll just bring everyone back onto webcam. Perfect. Welcome back, everyone. First question here, we'll start off with, for both of you perhaps, the videos that you've shown are really stable. How do you stabilize the setup, and can you give a sense of what the total time captured in the videos you presented was?
Yeah, so we don't stabilize the recordings, actually. I think they're quite stable as they are. Or maybe we would, yeah, when there's a lot of movement, we would stabilize with very simple algorithm. But during the recordings, or the recordings that I showed here, is not, they were not stabilized. I think, yeah, I think it's possible to get data for I mean, the entire experiment, but the imaging acquisitions that I showed was like a few minutes long, 2 minutes long max. But that would be based on how long it's advisable to continuously do two photon imaging,
right?
More than the setup as such.
Yes, I would actually say something similar to what Barbara has said. I think it is very important during the implantation step, right? So to just make sure that head post is really well positioned and fixed to the skull. And I have to say that I am really sort of a novice in these kind of things, but I've done the surgeries myself, and we had great support from Neurotar. I actually want to send hi to Leo Kirog, who's been great. They're always very available. The protocols were available to us, and so there is no image stabilization, although there are obviously digital algorithms that you can use to stabilize when things are not perfect. So yes, so that's one thing that I would say is that really these protocols are very mature in terms of preparing the animals for these experiments. And the videos that I showed are for the entire duration of the condition, which is 300 seconds, and are presented at 30 frames per second.
Okay, fantastic.
Yeah, I actually want to modify my answer a little bit. I'm sorry, because I have some videos where I didn't show raw data, where I show processed data, relative, like normalized calcium activity to the optogenetic stimulation, and that, of course, was image stabilized first, and images was normalized, and all this sort of thing. So and it was an average of several stimulations, and so on and so on. So it depends, but the raw images of mouse running and the calcium activity there, that was not stabilized.
Okay, great. Next question here. Barbara, did you look into more detail at the calcium signal spread in single astrocytes? And also, are there subtypes of astrocytes coupled to different interneurons?
So I think that is really two questions. So I would say, no, I did not study the spread of calcium activity, and you can do that using an algorithm called AQUA. But I used an algorithm initially. Used an algorithm very similar to AQUA, but I found that it's picking up the bigger responses, and the responses that I have to my interneuron stimulation is actually quite widespread, but puncture-like small, right? So to pick them up, it was actually more simple to just take field of view calcium signal. But in that way, of course, I wouldn't have any information on the spread. But that being said, the spread of calcium in astrocyte, it seemed to be more in the later stage of the calcium activity, where it was fluctuating, that there was a movement of calcium within the cell. With regards to subtype of astrocyte, I don't know that there is any knowledge about its specific connections to different sort of interneurons, and I did not look into that. But I think it's down the line, of course, something which we need to know.
Okay, great. Thanks. Great answer. Next question here for Adrián. And the question is, the calcium activity looks like it comes from the tissue in general. And would there be other cell types expressing this calcium indicator?
That's a very good question. And I would actually add another potential alternative for why the signal is coming from the tissue. And remember that even though the two-photon excitation is very focal, right? The size of that optical slice, right, is important. I have to mention a detail about the way that the data is acquired. We are not using very high numerical aperture objectives here. We are using a 20x, but I think the numerical aperture is about 0.5. So the optical slice is not as thin as you can achieve with higher numerical aperture objectives. So that's one possibility. That's also why we decided to compare region of interest changes, fluctuations in fluorescence with the entire frame. And when we look at the time course of these different, the global and the sort of the more local signal, we see differences between them. So that it's a very interesting question. So I think the contribution from the tissue is probably coming from other vessels that are within the excitation volume, and you are very good in asking about expression in other types. We assume that CDH5 is mostly expressed in endothelial cells, but I believe there are other cells in the organism that also express CDH5. When we do histology and we look at endothelial cell markers, we see a very good sort of colocalization of GCAMP expression and markers for endothelial cells. And we don't see much evidence that there are other cell types. We have tried neurons, we have tried microglia, we have tried, I think, astrocyte markers. And it's mostly, it's not a very extensive comparison we've done. But there are some movies that I didn't have time to show that are also very interesting, where we see little tiny cells sort of crawling along the inside of vessels that I have no idea what they are. But they are very interesting because sometimes they make it into the brain. I don't know if they actually came from the bloodstream or if they were already present in the brain. Again, it's anecdotal, but that's my experience in running the experiments. And unfortunately, I cannot entirely exclude that many of the changes in fluorescence we detect are from these other cells that are in vivo. We have tried to choose volumes, imaging, series where there's mostly what looks like blood vessels and not these other tiny cells. So that's what I have from my experience doing these experiments.
Okay, great answer. You've presented a mystery now, so people will be curious to find out what these cells are, these moving cells. I think maybe we'll try and squeeze in two last quick questions, one for each of you. First, for Adrián, for how long after recovery did you have reduced synchronicity between endothelial cell calcium signals and vessel dilations? So I guess how long did the effect persist?
Well, so what I showed you is what we did, right? So basically it's 5 minutes after maintenance that we let animals recover, and during those 5 minutes we've noticed that even though you can see that the animal the animal's awareness, right, comes back, and they start whisking, maybe they start moving. This is the period that we imaged, and we wonder about how long does this last, and we haven't really done a systematic study to see how long. But I have to say that several of the animals that were in the study were imaged several times, 2 or 3 times. So we haven't really analyzed that data to see, how, there are long-lasting effects, but the action in our hands, because there are days between imaging sessions, we cannot really tell a difference between the first session and the second or the third imaging session. So, of course, we didn't design a specific test for to do this, but it's a good question. We don't know how long, what is the time course of this disruption.
There was this paper coming out, I think it was of the Bruno Weber group a couple of years back. They said 20 minutes after isoflurane.
Okay.
But since you don't record for that, you would know. I don't know. I'm not. I hope I remember correctly here.
Okay, great. Maybe we can add some links into the handouts and resources. Okay, maybe last question, just in the interest of time, for you, Barbara. Does parvalbumin interneuron stimulation always induce vasodilation?
Yeah, so there are some. I mean, most studies that I've come across, they do see a blood flow response to a optogenetic PV stimulation. But there are a few studies where they stimulate maybe a bit more superficial or differently, where they actually see a reduction in the blood flow. So it's not a very simple stimulation paradigm, I would say. So I can only lean on to that it does happen for others and always for us. So in that context, it does. And of course, we check that it's not the optogenetic stimulation, the light in itself that's doing this dilation. It does seem to be channelrhodopsin specific. So yeah, that's all I can say for that. Yeah.
Okay. Yeah, fantastic. In the interest of time, I think we'll have to wrap things up there. But I'd just really like to thank you both for your time and wonderful presentations today. It's been an absolute pleasure having you with us.
Yeah, thank you, Andrew, and thank you. It was really nice to see your talk, Barbara, and to meet you.
You too, yeah.
To see you at a conference or something.
Yes.
All right, wonderful. Thank you both.
Okay, thank you. Take care.
Bye-bye.
Bye-bye.
I'd like to also say a big thank you to our audience. We certainly appreciate your participation and hope that you enjoyed your experience here today. Just before you go, we'd love to get your feedback on our survey. We definitely read all of your responses, and you can fill that out here. And finally, a huge thank you again to Neurotar for sponsoring today's event. And remember, if you'd like to meet the team in person, you'll find them at the upcoming SFN meeting in San Diego at booth number 2036. So just a quick reminder that a recording of today's webinar will be available in the future, and you will receive an email giving you access to that recording. And finally, if you do still have questions, you can continue to submit them using the Q&A panel, and we'll be sure to pass them on to Adrián and Barbara. And in closing, we hope you enjoyed this Inside Scientific webinar sponsored by Neurotar, and we hope to see you again with us the next time. Thanks very much, everybody.